Hello students, welcome to ICSE Board Class 10 Biology episode 1.3.1. In the previous episodes we have already seen about the outline of the subject as well as we have started with the chapter unit 1, chapter 1, that is cell division and the structure of chromosome. In the previous episode we have also seen about the outline related to the cell, definition and the functions of the cell organelles. You have to keep in mind the functions of cell organelles that are likely to be asked in the board examination is Golgi apparatus and endoplasmic reticulum. In this session, we are going to talk further related to cell division. So we are going to see cell division, part 2, structure of the chromosome as well. So what topics we are going to cover? We are going to cover introduction, need for the new cells or the reasons for the growth and development, cell cycle, interphase, cell division, types of cell division, mitosis, meiosis, structure of chromosome and summary. We are not going to complete all of these things in this session, but we are going to break it down into parts. So first of all, introduction. As you know, cell is a basic structural functional unit of life. We have seen this one in the previous episode as well as you are learning from class 5. Every cell has a tendency to grow and develop. That means when I talk about every cell, that is prokaryote as well as eukaryote. Prokaryotes are unicellular. There are unicellular eukaryotes as well as multicellular eukaryotes. Every cell has a tendency to grow and develop. The cell organizes itself according to its function and abilities. This is basically seen in multicellular organism. As for example, sperm and ovum combines to form a single cell called zygote by the process of fertilization. Zygote is a single cell. It divides and multiplies further from stage 1 cell to multicellular organism, and it is through 1 cell, 2 cell, 4 cell stage, then 8 cell stage, 16 cell stage, morula, blastula, gastrula. Likewise, it's going to go under the formation of organs. But during the gastrulation, the cells are numerous and they are going to arrange in 3 layers. That's why multicellular are coming under the title triploblastic. There are 3 germinal layers, outer one, middle one and the inner one. Outer one is called ectoderm, inner one is called endoderm and the middle one is called mesoderm. We'll study all these things in the chapter of reproduction. Function of a cell is to bring growth and development in the organism, do various metabolic activities to provide energy to it for living and help in sustained life. That means a cell is helping in what? Growth and development. A single cell divides, grows, divides, develop. So development of the organism is taking place. So it is cell growth and development, becomes mature, cell division occurs and it keeps on multiplying and the cycle continues. As well as it also helps in doing metabolic activities so that the energy is provided to it to sustain life. So this is all about the introduction. Let us move on. Why do we require to have new cells, or what is the importance of cell division? The importance of cell divisions are as follows: growth, replacement, repair, reproduction and regeneration. You can see G and there are 4 R's. So you can say G 4 R's. As well as the important one is to sustain life. Let us begin. Growth, it increases in the size and the development of organs and finally the organisms. So it helps in the increase in the size. Baby is small and it starts slowly growing and the height increases, organ develops, hand becomes longer, hind limbs becomes larger, the face changes. That is what? Growth and development. Number 2, replacement. It helps in replacing the old and worn out cells with the new ones. Students, whenever you are taking bath and wiping out your body with the towel, then what happens? Millions of cells are falling off. Then what is happening there? The new ones are being added, and that is with what? With the help of replacement, with the help of cell division. So cell division helps in growth, helps in replacement, and the number 3 is repairing. It helps in repairing damaged cells. If someone gets injured, then what happens? The new cells are formed. Repairing of the damaged cell is taking place. That is very important for our body. Reproduction, it acts as a mode of reproduction in the lower organisms like amoeba, paramecium and all. It is helping in what? In the reproduction So from one amoeba, two amoebas are produced by binary fission. So binary fission is a mode of asexual reproduction in lower organisms. The same thing happens with the paramecium and other prokaryotes. So reproduction is due to cell division in lower organism. The fifth one is regeneration. You might would have seen lizard's tail. If the tail of the lizard gets lost or cut, a new tail is formed. Salamander, starfish, hydra, if their arms are lost, accident or by fighting with other organisms or catching prey or hiding from their enemies, and if they lost any of its arms, then the new arms are regenerated. So that is a regeneration, helping in certain animals to develop their organs. So what is the importance of cell division? The importance of cell division is to sustain life, helps in growth, replacement, repair, reproduction and regeneration. Let us come to the another topic, that is cell cycle. This is the main topic, that is now our chapter starts from here. Cell cycle. See, the word cycle is used, that means there must be sequential events going in the cell. So to divide, each new cell has to undergo the phase of growth and development. So a newborn cell is there, it undergoes cell division, but before undergoing the process of cell division, it requires to grow and develop. That means growth and development is the word related to maturity. That means the cell requires to become mature. Once it becomes mature, then only it undergoes the cell division. It is after this phase that the cell attains enough maturity. So growth and development is taking place. That means the cell is becoming more and more mature and can complete all the metabolic processes before entering the phase of cell division. So whatever metabolic processes are there, it is been completed. Energy is provided, growth and development is taken place, maturity is there, and then it enters cell division. So this growth and development phase, which is termed as interphase, is also known as preparatory phase. So this part is what? Preparatory phase. Which one? The growth and development one, that is the interphase, is also known as preparatory phase. Which one? Which part I am talking about? I am talking about the growth and development. So I am talking about growth and development. So growth and development, preparatory phase, or you can say interphase. All these are same. So which part of the cell cycle is known as preparatory phase? Interphase. Growth and development phase of a cell cycle is also known as interphase or preparatory phase. I hope you understood that one. And it is afterwards, after preparation, after interphase, it undergoes cell division. So the cell cycle can be defined as a sequential events occurring in the cell to grow and to develop, to become mature, to undergo cell division. So it is pretty simple that a cell wants to go to process of cell division. But how it undergoes the process of cell division? By becoming mature. How it becomes mature? By attaining growth and development, by achieving growth and development. So once it takes growth and development, it becomes mature, and then it undergoes cell division. And this thing continues with the other cells also. So from one cell, two cells are formed. From those two cells, again 4 cells are formed. And from one cell to 2 cells, how it has taken place? By becoming mature. As for example, a plant becomes a tree. There is a seed. I am sowing it under the ground. Next day it is not going to grow into a tree. It is going to go through various stages. And once it becomes mature, it will start giving fruits. So that's what it is. Puppies are not going to become dog over a night. So it has to undergo the process of growth and development. It requires to become mature. Once it becomes mature, it starts reproducing. Same thing happens with the body cells. I hope you understood the meaning of cell cycle. Sequential events occurring in the cell to grow and develop, to become mature, to undergo cell division is known as cell cycle. So that means cell cycle I can write the preparatory phase. The preparatory phase is what? Interphase. The preparatory phase is the interphase. Interphase is known as preparatory phase or growth and development phase. I hope you understood. And then it is followed by the cell division. So interphase plus cell division is equal to cell cycle. So this is how it looks. So cell cycle. So if you see, yes, from here, this part, the cell begins to grow, and then it divides into 2. and then again this new cell which is formed, it is going to go again back to the cycle. So this is how it takes place. So first of all, cell, it undergoes the process of growth and development. That means the growth and development is an interface. So from here, from this part to this part, it is an interface stage, and then here it is written M, that means it is the part of cell division. So this is a part of cell division. We'll see this one in the further episode, but let us finish up with the first part of the cell cycle, that is interface. So this is how it looks like. So here let us see this diagram. So here this is the parent cell. Parent cell has undergo the process of growth and development, that is interface. Once the cell has entered interface, it becomes mature. When the cell attains maturity, it undergoes the process of cell division and forms two daughter cells. This daughter cell grows and develops, so it becomes again undergoes the process of interface. It attains maturity, cell division, and then two daughter cells. Same thing with this one. So I hope you understood. This part. So it is what it is. Let us see once again. First of all, cell. Here, first of all, cell, growth and development. What is the growth and development of a cell cycle? Known as interface. Maturity takes place. Thus, this becomes the mother cell. Mother cell divides into two daughter cells. These two daughter cells undergo the process of growth and development. Again, growth and development takes place, so it becomes the cell which is equivalent to its mother cell. That means it is gaining the size of its parent, of its mother, and then again cell division takes place and two daughter cells are formed. Again these daughter cells will go further, and the cycle keeps on continuing. I hope you understood this one. So let us see here. Here GD means what? Growth and development. To become mature, that is interface. After maturity, the cell enters cell division, producing two daughter cells which further undergo growth and development to attain maturity, and the cell cycle continues. I hope you understood this diagram. So let us see the definition once again. The sequential event, so you have to write the word. So this is the most important. You need to write sequential event occurring in the cell to grow and develop to become mature to undergo cell division is known as cell cycle. So this is the definition of cell cycle. So cell cycle means what? Interface plus cell division. But one thing is there, that in cell division, this part. One thing is that in cell division, it depends upon the cell location. Cell division will undergo either mitosis, either mitosis or meiosis. Mitosis, then it is a somatic body cell, and meiosis, then it is a reproductive germ cell. When I say somatic body cell, that means it is related to skin, tongue, and meiosis related to the reproductive germ cell. That is, for plants it is androecium and gynoecium, and for humans or for mammals it is sperm cell. Sperm cell. Yes, it is sperm cell and ovary cells, or you can say germinal layer cells. Let us see the process of interface. Not the process, but the phase of interface. So how the process this is taking place here. Interface is the phase of cell cycle where cell undergoes growth and development and prepare itself to enter cell division. It is a long phase of about 17 to 18 hours, and it takes place in the body somatic cell. I have written body somatic cell, but it is any cell, but it is taking place in the body somatic cell also. Before dividing, each cell has to attain the same size as that of mother cell. That what we have seen in the previous slide. And here there are three sub stages. Which are the three sub stages? These are the three sub stages: G1 phase, S phase, and G2 phase. So these are the three phases of interface. So let us see one by one. So first of all, G1 phase. G1 phase is also known as gap 1 phase or growth 1 phase. It is the initial growth phase, first growth phase of the cell, where the amount of cytoplasm increases. So here amount of cytoplasm increases. Cytoplasmic organelles like mitochondria, endoplasmic reticulum replicates. Cell size increases. Cell organelle develops and starts replicating. Cytoplasm of the cell increases in the size. Cell volume increases. DNA content remains same. It develops, but it remains same. This is the most important part. In which phase of interface DNA content remains same? In G1 phase. So these are the characteristics of G1 phase. Now there are certain cells which Now the cells which has everything, like the cell which has cell organelle, cytoplasm is very well developed, DNA content is very well developed, then only it will enter into the another phase. But what about the cells which do not have cell organelles? Where they will go? So the cells which do not have cell organelles, like RBC, like neurons, they don't have cell organelles. They lack cell organelles. As for example, RBC does not have cell organelles and neurons does not have centrioles. Then what about such type of cells? Such type of cells will remain in G0 phase throughout its life. So whole life RBC and neuron is going to stay there in G0 phase. It is not going to move from G1 to S phase. It's not going to move from G1 to S phase. Why? Because there are no cell organelles. There is no centriole. Cell organelles are not there in RBC and centrioles are not there in neurons. So such type of cells, these are the example that I have mentioned where those cells remains in a G0 phase because they don't have cell organelles. Why RBC does not undergo cell division? Because it lacks cell organelles. Why neurons does not undergo cell division? Because it lacks centrioles. Let us move on further. The cells which have cell organelles, completely well developed cell organelles and cytoplasm, are moving further into S phase. S phase is also known as synthesis phase. Chromosomes are duplicated by the synthesis of more DNA. DNA replication means copy, duplication takes place. Histone proteins are synthesized. Don't worry about histone proteins right now. We'll see in the next chapter. Histone proteins are synthesized. Other proteins are also synthesized, which are useful in the next phase of interphase, that is G2 phase. So whatever proteins are synthesized in this phase is going to be useful in G2 phase. So let us see G2 phase. The cell which has already entered into S phase will definitely enter into G2 phase. But the cell which has entered G1 phase is not sure that it will enter into S phase, like RBC and neurons, because they don't have cell organelles, so they are lagging behind and they are going to stay there in G0 phase. But the cells which have complete structure, complete development, cell organelles and cytoplasm and volume and DNA duplication, everything is done, then it is going to enter into S phase, and from S phase it is going to enter into G2 phase. So it is sure that the cell which has entered into S phase is definitely going to enter into G2 phase. G2 phase is shorter. Proteins are required for the cell division are synthesized here. Cell has increased size. Cell prepare itself to enter for the next cell division. Here the nucleus enlarges. Definite number of chromatin and chromosomes are becoming visible. Right? DNA material, nuclear material, DNA is becoming double. So these are the things that you have to mention for the G2 phase. Centrosome duplicates and centriole starts moving towards the pole. So this is also one of the important feature. Centrosome duplicates and centriole moves towards the pole for the formation of ester rays. Ester rays are actually helping in the formation of spindle fibers later on. Okay, let us move on. Different types of RNA, hnRNA, that is heteronuclear RNA, mRNA, rRNA, tRNA are synthesized. So these are the characteristics of G2 phase. Characteristics of G1 phase. 1 mark question. DNA content remains same, that is 1 mark question. Examples of the cells that remain in G0 phase and why, that is 1 mark question. Where does the duplication of DNA takes place, that is 1 mark question. State the characteristics of G2 phase, 1 mark question. Difference between G1 and G2, 1 mark question. And here this is the most important concept of chromatin, chromosome and chromatids. Chromatin, if you see the structure of chromatin, chromosomes and chromatids. Chromatin, network like structure. Chromosomes, highly coiled structure with the visibility of centromere. So here this is the nuclear membrane. 1 is a nuclear membrane, 2 is the dot holding the arms of chromosomes known as centromere, and duplicate arms of chromosomes is known as chromatids. So here definition is there, and I have shown the diagram related to chromatin, chromosomes and chromatids. So please don't get yourself confused. So these are the number 3. You see the number 3 here, this one number 3. So this one, if I say I will label number 1, this part and this part are the duplicate arms of chromosome. This part and this part are the duplicate arms of chromosome. Same here. While this is chromosomes, the duplicate arms of chromosomes are known as chromatids. Let us move further. So this stage is called interphase, right? So which stage is called interphase? G1, S and G2 is known as interphase. It is also called resting phase. So here it is, another name is resting phase. Resting phase or resting stage, while in fact it is not resting, it is highly metabolically active stage. Hence it is not been appropriate to mention as a resting stage. So it is not good to say resting stage. It is good to say metabolic active stage, or you can say preparatory stage, because cell is preparing itself to enter cell division. So this is how it looks like afterwards. So here if you see here G1, so this is the G1 stage, this is the S phase, that is duplication of DNA is taking place. This is the G2 phase, final touch is taking place, and these are the 4 stages of mitosis that what we are going to study in the next episode. Let us move further. So here what happens in the interphase? Cell size increases, nucleus of dividing cell enlarges to its maximum size, chromatin turns into chromosomes while visibility of centromere and starts replicating. That is, a copy of each chromosome is created to form chromatids but still remain attached with the centromere. Nucleolus is very clear, as well as cell organelles starts forming copies. The energy for the cell division is stored as proteins, and certain substances necessary for initiation of cell division are also formed. So these are the things that is taking place in the interphase. So I hope you understood the process of interphase, or you can say the stages of interphase G1, S and G2. I hope you understood the other characteristics as well as chromatin, chromosomes and chromatids. If you have any doubts, please let me know in the comment box. We'll see the rest part of the chapter in the next episode, because I wanted to teach you step by step in a proper manner. Otherwise I can finish it up within another of 40 minutes or 45 minutes. I can finish up the whole chapter, but I don't want to do that. So I will see you in the next episode. If you have any doubt or something, please let me know. Thanks very much folks about the episode.